The second lecture is on the discovery of receptors for touch by Dr. Ardem Patapoutian. Ardem Patapoutian is professor in the Doris Neuroscience Center at Scripps Research in La Jolla, California, and Howard Hughes Medical Institute investigator. Professor Patapoutian was born to an Armenian family in Beirut, Lebanon, where he attended elementary and high schools. After graduating from high school, he attended the American University of Beirut for 1 year before immigrating to the United States of America in 1986. Professor Patapoutian received his bachelor's degree in cell and developmental biology from the University of California, Los Angeles in 1990, and he obtained his doctoral degree in biology from California Institute of Technology in 1996, where he worked with Barbara Wold focusing on muscle differentiation. Professor Patapoutian performed his postdoctoral training in the laboratory of Louis Reichardt at the University of California, San Francisco, where he worked on the developmental program driving subtype specificity of somatosensory neurons for touch and pain. Ardem Patapoutian joined the faculty in the Scripps Research in 2000, and he also held a position at the Genomics Institute of the Novartis Research Foundation between 2000 and 2014. Ardem Patapoutian is a member of the National Academy of Sciences and a fellow of the American Association of Advancement of Science. Professor Ardem Patapoutian will now deliver his Nobel lecture entitled, "How Do You Feel? The Molecules That Sense Touch."
It has been such an amazing experience to share the 2021 Nobel Prize in Physiology or Medicine with David Julius. I thank the Nobel Assembly, members of the Nobel Committee for Physiology or Medicine, and the scientists who have supported our nomination. I'm grateful that basic research is being recognized in such a way as biomedical laboratories around the world not just advance our understanding of life, but are the nurseries for future medicines. Our perception of reality is defined by our senses. We have learned quite a bit over the past decades about how we smell, taste, and see. We know much less about the sense of touch, which gathers mechanical, thermal, and chemical information from peripheral tissues such as the skin and translates into an electrical signal that neurons understand. More generally, most cells in our bodies communicate through chemicals, but cells also respond to pressure, and how this is accomplished has remained unknown. So I'm here to tell you that your sense of touch is amazing. Human tactile discrimination extends into the nanoscale. You can sense indentations 500 times thinner than a human hair. Beyond this, we rely on affective touch for our emotional health, and for the related sense of proprioception allows us to know where our fingers and arms are in space without looking at them. And this allows you to perform complex tasks such as walking or playing a musical instrument, beautifully demonstrated here by Yo-Yo Ma and Emmanuel Axe. But I don't know of any other image that highlights the power of touch more than this one. So this boy is visiting the White House and tells President Obama that his friends tell him that his hair looks like the president's. Mr. Obama lowers his head and says, "Why don't you touch it?" Of course, we're not just talking about sensing texture of the hair, but the intimate, affective nature of touch. We often say seeing is believing, but in this case, touching is believing. So for mechanosensation, the flip side of touch is, of course, pain. Acute pain that warns us of danger in our environment is essential for survival. Chronic pain, when the noxious stimulus is no longer there, but pain persists, remains a major unmet medical need. For the neurons responsible for detecting mechanosensory stimuli relevant for touch, proprioception, and pain, mainly reside in dorsal root ganglia, or DRGs, a group of sensory neurons present just dorsal to the spinal cord. So they gather information from the periphery and send information to the spinal cord and the brain. DRG neurons are very heterogeneous. Some are specialized to sense temperature and pain, but low-threshold mechanoreceptors respond to touch and proprioception. These neurons are also characterized by their peripheral targets. For example, proprioceptors innervate muscle spindles, whereas various neural endings that are found in the skin are responsible for the sense of touch. Here I'm simplifying sensory receptors responsible for touch, but different fiber types, such as Merkel cell neurite complexes and Meissner corpuscles, play unique roles for our sense of touch. And the labs of David Ginty, Ellen Lumpkin, and many others have contributed to a deeper understanding of this process. In addition, many DRG neurons project to internal organs, such as the bladder, and are involved in a process called interoception that I will talk about later. So how do you sense a gentle breeze or a cactus pricking your finger? We have known for quite a while that somatosensory neurons, for example at your fingertips, express specialized ion channels that are directly activated by physical forces. So over the last 25 years, work led by David Julius, my lab and others have identified trip channels required for thermosensation and pain. However, just 12 years ago, the identity of mechanically activated channels for touch and pain, or really any other biological process that depends on pressure sensing, had remained elusive. For those who are not molecular biology experts, I wanted to provide a scale of how small these sensors are that we're looking for. So a grain of rice is 500 times bigger than a cell. Here's an average cell, about 10 micrometers. An ion channel is about 100,000 times smaller than a cell. They would be sitting on the plasma membrane, but they're too small to be seen with this scale. So what do we know about these elusive mechanically activated ion channels? Hudspeth and colleagues were the first to demonstrate their existence and importance. They showed that signaling was too fast for hearing to be explained by a second messenger system. So they hypothesized the direct activation of a mechanically activated channel to be involved in hearing. Later on, Sachs and group showed that this type of stretch-activated currents are present in many different cell types. So how about the identity of these ion channels? So mechanically activated channels have been discovered in many species, including bacteria, worms, and flies, and shown to play important mechanosensory roles. However, either these sensors did not have homologs in mammals, or a direct role for them in mechanosensation was not forthcoming. Interestingly, mammalian mechanosensitive ion channels that conduct potassium have been identified. However, this could not be the touch sensors, for example, as their activation would inhibit rather than activate neuronal firing. So it was not till 2010 that my lab identified Piezo1 and 2, non-selective cation channels from mammalian cells. These are the ion channels I will discuss today. So why has it taken so long to identify these elusive mechanosensors? It would help, perhaps, to look back how other sensors were identified. So the first transduction molecules identified was for vision. Rhodopsin, a GPCR, converts light into electrical signal. Now, 50% of the disc membrane area of a rod outer segment is made out of rhodopsin. And this availability in large amounts enabled biochemical approaches to purify and identify the sensor. It was known that G protein signaling was also involved in olfaction and taste. And indeed, Axel and Buck and Zucker and Riba identified olfactory and taste receptors by homology to rhodopsin family of GPCRs. However, the mechanosensors we're looking for are known to be ion channels, and unlike GPCRs, these channels do not have robust homology among each other. Therefore, we thought a functional genomic screen was the best approach to identify them. So today, I will tell you about how we identified piezo ion channels, and then I will focus on the role of neuronal Piezo2 in somatosensation and interoception. Finally, I will touch upon the role of Piezo1 in non-neuronal cells. This is Bertrand Coste, a postdoctoral fellow in my lab, who came specifically to identify the molecular identity of these mechanosensors. And now he's a faculty member in Marseille, France. So Bertrand adopted a simple cellular assay to assess mechanically activated channels. So while recording from a cell, he would come with the glass probe and push the cell. And if this cell expresses a mechanically activated channel, he could record such currents. He then took a reductionist approach and to try to find this channel from a simple to work with cell line, so that once we identified the channel, later in vivo work would tell us which physiological settings it's playing an important role in. So he screened several cell lines and found that Neuro2A cells have robust mechanically activated currents. And then he used various bioinformatic tools to come up with about 300 candidates that could be this channel. For example, they would have to be multi-transmembrane domain proteins and would be expressed in Neuro2A cells. And then he would knock them down one at a time using an RNAi screen to find the molecule responsible. So this is the screen that he performed. He would knock down each candidate represented by a single data point, one by one, and he would record maximal mechanically activated currents. To get these kind of error bars for each data point, he spent about 2 to 3 days on each candidate. So after almost a whole year of just getting negative data, he finally found that knocking down candidate number 72 had a profound effect on these mechanically activated currents. So this candidate was called unknown family 38 member A. He quickly renamed it Piezo1 from the Greek piezi, meaning pressure. So we got really excited then when Bertrand got these results. So he was able to clone the full-length Piezo1, and when he expressed them in naive cells, such as HEC293 cells, he could see robust nanoamp level currents. So this is showing one such recording where the more you push on the cell, you see larger and larger currents. And this is the quantitation for those results. So Piezo1 has a sister. We named it Piezo2, and it's about 40% identical at the amino acid level. And it's also mechanically activated, albeit with a slightly different inactivation kinetics. So one of the unique properties of piezos is that they're very large molecules. Mouse Piezo1 is about 2,500 amino acids and has many transmembrane domains. So all ion channels have to have domains that traverse the membrane, but normally ion channels would have, on average, 2 to 6 such transmembrane domains. Here you can see in orange bars that piezo has 38. That is actually more transmembrane domains than any other protein in your genome. In addition, we now know that piezo comes together as a homotrimer, forming a complex that is almost 1 million Daltons. So Kei Satome in the lab, together with Ward Lab here at Scripps, used cryo-electron microscopy to solve the structure of Piezo1. Similar structures were solved by McKinnon and Zhao labs. So you can see the 3 protomers here shown in different colors. They all come together at the central C-terminus where the pore is. The rest of the protein, you can see each ribbon is a transmembrane domain, extends into the plasma membrane. And they extend into the plasma membrane, presumably acting as membrane tension sensors. You will immediately notice that the whole structure has an unusual upward bend to it. And the current hypothesis, first proposed by Rod McKinnon, is that these channels sit in grooves within the plasma membrane, and that any tension or stretch of the membrane will cause flattening of piezo structure and hence activation. So to complement our in vitro work on the structure and function of these ion channels, we have spent considerable amount of time elucidating their function in vivo. So both in mouse models as well as from human clinical data, we have been exploring the role of piezo ion channels and asking what happens if piezo is not present, loss of function studies, or when we have too much piezo signaling, or gain of function studies. We were very interested when we found that Piezo2 is robustly expressed in the sensory neurons of the dorsal root ganglia. These are the neurons I told you about that are required for touch and pain. This is an RNA scope, RNA in situ hybridization, that shows in brown expression of Piezo2 in a large subset of these sensory neurons. So we have made transgenic mice that do not express piezo and have done many experiments to show that Piezo2 is the principal sensor for touch. I just wanted to show one very early experiment that we did. So these are wild-type animals, and we've put a tape on their back. As you can see, they don't like it and try to remove it. Piezo2 knockout animals, on the other hand, as you can see, they don't seem to be bothered by this at all. So this work was carried out by Sanjeev Ranade and Sung Woo in the lab, collaboration with Lumpkin and Lewin labs. So I mentioned proprioception before. I think this is perhaps your most important sense, and yet most people don't know that they have it. The sense of where your body is in space is based on mechanosensory neurons that innervate muscle and tendons. Thanks to this sense, you are coordinated, can close your eyes and touch your nose, and with a lot of practice, might be able to do things like this. So is Piezo2 required for proprioception? Work done by Sung Woo in collaboration with Wilkinson and Jessel labs also showed that Piezo2 in the mouse is required for proprioception. Here is an animal that has no Piezo2 mid-thoracic and lower. You can see that this mouse lacks coordination in its hind legs, a sign of lack of proprioception. For all behavioral tests that we do, we confirm the role of Piezo2 in transduction pathway by using ex vivo electrophysiology. In this example, Wilkinson lab can stretch muscles while recording neuronal activity from proprioceptors. As you can see, in wild-type animals, as you stretch the muscle, you see massive increase in neuronal activity. In contrast, in the knockouts, we hardly see any activity. Interestingly, the labs of Chesler and Bonneman at NIH identified patients with loss-of-function mutations in Piezo2. And very interestingly, they have the same deficits in touch and proprioception. So in this video, a test for proprioception, one of these individuals is asked to walk in a straight line while blindfolded and harnessed for their own protection. As you can see, she has a very difficult time performing this task, something that you and I can easily do. So how about pain? We know both in mouse and humans that acute pain is relatively normal, but we wanted to ask about tactile allodynia. So this tactile allodynia is a painful sensation that is caused by an innocuous stimulus such as touch. You might experience this after injury or just after sunburn, but this is also a phenotype experienced by patients who suffer from neuropathic pain. So Shweta Murthy in my lab, as well as Alex Chester's lab, demonstrated that Piezo2 is essential for tactile allodynia, both in mice and in men, opening a possibility that blocking Piezo2 in some instances could be clinically relevant for people suffering from chronic pain. So the requirement of Piezo2 in touch, proprioception, and pain entails sensing from peripheral tissues such as skin and muscle. But mechanical force transduction is also relevant for internal organs. So although DRGs do innervate internal organs, there's also a whole other sensory system dedicated to this. So the cranial ganglia of nodos through the vagus nerve innervates internal organs and gets mechanical and chemical information from them, including lung, the heart, stomach and the gut. Both the mechanism of mechanotransduction, as well as, in some cases, the precise physiological role of mechanotransduction in these tissues is not understood. Over the last few years, work done by Keiko Nonomura, Sung Woo, Weizheng Zhang, and Kara Marshall, and in collaboration with Liberless Lab and Chester Labs, we have begun to investigate the role of piezos in interoception. For example, lung stretch is sensed by these sensory neurons, and this information is used to control breathing rate. Piezo2 plays an important role in this process. Although you're not conscious of this, there are specialized neurons that sense pressure in your aortic arch, and this information is used to keep blood pressure constant. Piezos are also required for these baroreceptors. On the other hand, you are very aware of bladder stretch as it alerts you when it's time to relieve yourself. Both in humans and in mouse, Piezo2 is required for this important sense. And indeed, we found that humans without Piezo2 plan to go to the restroom on a schedule to avoid accidents. And I wanted to highlight that the role of mechanosensation in interoception is an active area of research. We are, for example, currently investigating the role of piezos in the gut. When you eat a large meal, you feel very full. That sounds very mechanical in nature. And yet, most of research on satiety is focused on nutrient sensing, for good reason. But we do not know how much stomach stretch contributes to how much you eat, when you eat, diabetes, obesity. We hope that studying these molecules will illuminate the role of mechanosensation in food intake and digestion. So Kara Marshall and Rocio Servin in the lab are currently investigating these related questions. So I've talked mainly about sensory neurons, but many other cell types experience, sense, and respond to mechanical force. For example, blood vessels experience shear force from blood flow, muscles get stretched, and many different cell types get mechanically indented. Not just sensory neurons, but for example, red blood cells that squeeze through capillaries half their diameter. And the mechanism of mechanotransduction in most of these tissues remain unknown. So Piezo1, the sister molecule of Piezo2, is widely expressed in many tissues, and our lab and many others have only now begun to explore the role in various biological processes and diseases. For example, we and others have shown that Piezo1 is required for blood vessel formation during development. Others have shown an important role of Piezo1 in regulating bone density. Very recently, together with Meda Patek's lab, we showed that Piezo1 in the skin is involved in wound healing. Finally, we and others have found various interesting and unexpected roles of Piezo1 in red and white blood cells. I will spend a few slides talking about this. So we have strong genetic evidence that Piezo1 plays an essential role in red blood cell volume regulation. So gain-of-function mutations in Piezo1, both in mice and in humans, that have a condition called hereditary xerocytosis, have dehydrated red blood cells, while loss-of-function mutations in mice causes overhydrated red blood cells. So the hypothesis for normal function of Piezo1 in red blood cells is that when they get squeezed through capillaries, the RBCs actually shrink to more easily navigate these tiny vessels. In addition, partly due to the red blood cell dehydration phenotype, Shangma in the lab showed that gain-of-function Piezo1 provides protection against malaria infection in mice. So the next question was, if excessive Piezo1 signaling in the red blood cells is indeed protective, are there Piezo1 gain-of-function mutations in regions that are endemic for malaria? We indeed found a subtle gain-of-function allele, E756 delete, a deletion of a single amino acid, that is very prevalent in African population. So this map shows the frequency of this allele. The wild-type copy is in light blue and the gain-of-function in purple. As you can see, E756 delete is almost non-existent in people of European ancestry, but prevalent in African population as well as in African Americans. And this allele is quite abundant. 1 in 3 people of African descent are heterozygous for this allele. That translates into about 400 million people carrying this allele, with about 40 million people being homozygous. Now, since this allele is a very subtle gain-of-function mutation, it does not by itself cause any disease, but we think it is very interesting to investigate the consequences of carrying this allele. And we are finding out that there might be both advantages and disadvantages of carrying this allele, perhaps similar to what is known about sickle cell disease. So the advantage, as I mentioned, is its potential to protect against malaria. This is mainly shown in mice, and more work needs to be performed to assess its relevance in humans. On the other hand, we have recently shown that too much Piezo1 signaling in macrophages causes increased phagocytosis and iron levels in the blood, which is not good, as iron overload is associated with many pathologies, including organ failure. I will show a single slide on this data. So Shangma received blood samples from more than 300 self-reported healthy African Americans, and this shows the distribution of E756 delete. As expected, about 1 in 3 have the gain-of-function allele, shown in green, and very few are homozygous in orange. However, when we look at clinically relevant markers of blood iron levels, transferrin saturation and ferritin, we notice that within individuals that carry high iron levels, we observe a higher percentage of E756 delete carriers. Indeed, with a more stringent assessment of increased iron levels, all 10 individuals had at least 1 copy of the gain-of-function allele. Now, Shang has shown that these E756 delete carriers are 12 times more likely to have high ferritin levels than non-carriers. Larger studies are, of course, required to assess the risk factor of individuals carrying this allele. And finally, for the future, we are eager to perform more genotype-phenotype association studies to find out other potential physiological processes that depend on mechanotransduction. So till now, I have focused on mammalian Piezo1 and 2. This slide shows, however, that piezos are conserved across evolution. So flies and worms have piezo, many unicellular organisms have piezo, even plants have piezo channel. Plants also experience mechanical force. For example, when roots grow and encounter a harder surface in the soil, they have to decide whether they go right through it or around it. Recently, Ali Reza Musavi in my lab showed that piezo in plants is expressed in root tips. So in Arabidopsis, he showed that without piezo, the ability of these roots to penetrate through harder surface is compromised. I find it absolutely fascinating that the same type of sensor that we use to sense touch is also used by plants to sense substrate stiffness. Evolution is indeed amazing. In conclusion, we have shown that Piezo2 is the principal mechanosensor for touch, proprioception, and many interoceptive functions. We have also shown that Piezo1 plays a predominant role in many non-neuronal cell types. Overall studies in both human and mouse genetics are providing a foothold into unexpected roles of mechanotransduction in physiology and disease, including surprising roles such as regulating blood iron levels. Finally, there are more mechanosensors to be found. For example, for noxious pain, we know that there's a separate channel that accounts for this function. However, its identity is unknown. So the field is ripe for many more exciting discoveries. I have many people to thank for their roles in my scientific journey. I fell in love with basic discovery research in Judy Laniel's lab at UCLA, learned how to ask important questions in Barbara Wald's lab at Caltech, was inspired by Lou Reichard at UCSF to take on high-risk endeavors, and discovered how to harness cutting-edge technologies from Pete Schultz at Scripps Research. Learning from all these mentors I have tried to build a laboratory environment at Scripps Research that emphasizes impact, collaboration, and collegiality. I hope they are having as much fun as I am while pursuing such science. And I'm exceedingly proud of how well they work with each other and with our expert colleagues around the world. So these are the names of all the scientists who have worked with me in the past 21 years. These include current members as well as alumni. Indeed, one of the greatest joys of doing science is to see the trainees blossom into independent thinkers, chasing important questions both in academia and industry. And of course, we could not have accomplished much without collaborators from laboratories across the United States and beyond. So the importance of funding in biomedical research cannot be overstated. Howard Hughes Medical Institute affords freedom to pursue major innovations and has fueled my research program. The National Institutes of Health has been vital to the very existence of basic biomedical research in the US. During our COVID pandemic, I sincerely hope that the United States, and indeed the world, has a renewed level of appreciation for the impact biology research has on human health. And I want to end with a personal note on this image with my son Luca and me, a few minutes after I received the call from Stockholm and while the official announcement was being made. I'm very grateful that my wife Nancy snapped this photo. I study the sense of touch, and this is indeed a very touching moment of pure joy. My family has been a profound source of happiness and the foundation of my ability to do science. And because this is 2021, with restricted in-person interactions due to the COVID pandemic, this photo perhaps took on added meaning as it spread the news of the prize. And through social media, I received messages from around the world. Indeed, many congratulations came from people of Armenia and Lebanon, where my ancestors and I were born. I am grateful that the Nobel Prize has reconnected me with my roots and highlighted how internationalism, diversity, and the exchange of ideas and people are so integral to progress. Thank you for the opportunity to share my science with you.
I guess you all share my sentiments. This was fascinating, it was touching, and it had a clear address to all of us. On behalf of Karolinska Institute, I would like to thank you both, Dr. Julius, Dr. Patapoutian, for your outstanding lectures here today. Through your discoveries about our ability to sense heat, cold, and touch, you have contributed greatly to our understanding of how we perceive the world around us, and I should add, the world within us. You have identified critical missing links in our understanding of the complex interplay between our senses and the environment. Knowledge that will benefit a wide range of areas in the realms of research and healthcare. So, we very much look forward to welcoming you both to Karolinska Institute and to our beautiful Aula Medica when circumstances allow, hopefully in a not too distant future. And to all of you who have joined us today here in this hall or on distance, thank you for your attention. I'm sure you have also found the lectures very rewarding, and I wish you a pleasant day and an inspiring Nobel week. Thank you very much for your attention.